M. Shalini Garu, scientist to Eurofins Lancaster Laboratories, Massachusetts, USA, and participants from various esteemed pharmacy colleges from all over India. I am delighted to share that our college VV Institute of Pharmaceutical Sciences was established in the year 2010 under the sponsorship of AANM and VVRSR Education Society, Gudlavalle, Krishna District, Andhra Pradesh. The institute currently offers both B.Pharm, M.Pharm, and PhD programs. And our college was approved by Pharmacy Council of India, New Delhi, and affiliated to JNT UK, Kakinada. And our college is accredited with NAC and certified with ISO 9001-2015 certification. It is with pride I announce that our institution is hosting this online faculty development program over 6 days, featuring 6 distinguished speakers from both national and international backgrounds. Objectives of this particular faculty development program. And the first thing is enhance awareness of emerging trends, to introduce faculty to cutting-edge developments in the pharmaceutical industry, such as artificial intelligence in drug discovery, personalized medicine, and biotech innovations. And second important objective is bridge academia industry gaps, to promote collaboration by aligning the academic research with real-world industrial applications and regulatory frameworks. And third one is promote research and innovation, to encourage faculty in engaging in the advanced research and incorporate innovative methodologies in their work. And fourth objective is curriculum modernization, to assist in updating academic curricula to reflect current industry trends and future job market requirements. Fifth objective is skill development, to strengthen the technical, analytical, and pedagogical skills in key areas like digital health, nanomedicine, pharmacogenomics, and drug delivery systems. And next, sixth objective is to foster the interdisciplinary collaborations, that is content pharmaceutical sciences with biotechnology, data science, and regulatory science, to encourage the interdisciplinary learning. And last objective is empower faculty as change agents, build leadership and innovation capabilities among faculty to lead institutional transformation in pharmaceutical sciences. I take this opportunity to express my heartfelt gratitude to the esteemed members of the management, Dr. V. Nageswara Garu, Chairman, Sri M. Srinivas Rao Garu, Co-Chairman, Sri V. Satyanarayana Rao Garu, Secretary Correspondent, Sri V. Ramakrishna Garu, Co-Secretary Correspondent, Sri V. Subba Rao Garu, President of Promoter Society, Sri K. V. Krishna Rao Garu, Vice President of Promoter Society of AANM and VVRS Education Society and VV Institute of Pharmaceutical Sciences for their unwavering support and vision for institutional excellence. I extend special thanks to Ms. V. Satyasri Garu, Associate Professor Head, Department of Pharmaceutical Chemistry, and convener for this particular FDP program, for her dedication and initiative in organizing this particular program. My sincere appreciation also goes to Dr. Sheikh Amina Biga, the coordinator of FDP's VVIPS, for her meticulous planning and coordination. I am confident that this FDP will build a remarkable and enriching experience for all attendees. It will be broaden perspectives on pharmaceutical innovations and applications and serve as a catalyst for academic and research excellence. I am pleased to inform you that we have a tremendous response in the program, with over 300 faculty participants registered for more than 50 reputed pharmacy institutions across India. And thank you all for your enthusiastic participation and keen scientific interest in drug discovery and development. I encourage all participants to actively engage in all 6 days of plenary sessions to maximize learning experience. On day 1, we are privileged to have Ms. M. Shalini Garu, scientist, Eurofins Lancaster Laboratories, United States of America, delivering her talk titled Innovations Unveiled, Novel Approaches to Drug Design and Delivery. Now, I hand over the session to Ms. B. Satyasri Garu to continue the program. And finally, I wish this particular FDP program a grand success. Thank you, one and all.
Thank you, sir, for your welcoming note. Now I request our faculty member G. Manasa, Assistant Professor, to introduce the speaker of today's section. Manasa.
Thank you, ma'am. Good morning to one and all. And I'm very much thankful to introduce our today's speaker, Ms. M. Shalini, currently working as scientist to clinical microbiologist in analytical research and development in Eurofins Lancaster Laboratories, USA. Ma'am completed her Bachelor of Pharmacy in Acharya Nagarjuna University, Guntur from the year 2017 to 2021. And ma'am completed Master of Science in Regulatory Affairs from Northeastern University, Boston from 2020 to 2023. And she advancing pharmaceutical quality through expertise in microbiology and regulatory strategy. Also efficient in clinical testing and regulatory excellence to help deliver safer therapies to the world. Ma'am proven ability to adhere to safety standards, execute CGMP procedures, support CAPA tasks, and maintain meticulous documentation. Skilled in chromatography, filtration, equipment troubleshooting, and audit preparation. Committed to continuous improvement, cross-functional collaborations, and operational excellence in fast-paced biotech environments. She organized environmental awareness campaigns and coordinated donation drives and mentored volunteers and developed collaborative initiatives with non-profit partners. Thank you.
Thank you, Manasa, for the introduction. Nice introduction. Now I request Shalini ma'am to deliver a talk on innovations unveiled, novel approaches to drug design and delivery. Shalini ma'am.
Thank you. Thank you so much for the introduction. Good morning, everyone. My name is Shalini Manchkalpudi. I'm currently working as scientist, as already introduced. With a strong background in pharmacy, microbiology, and regulatory affairs, I have had the privilege of working on advanced testing methods and regulatory process that support pharmaceutical innovation, especially in vaccines and biologics. Today, I will be walking through transformative innovations in drug design and delivery. We will be covering topics like artificial intelligence in drug discovery, the promise of personalized medicine, role of mRNA and nanotechnology, regulatory considerations, and ethical implications. Throughout this talk, we will share the real world case studies and current lab practices, making this session as insightful as possible. I'll also engage with few interactive questions to keep the session dynamic. So let's delve into the session. I hope my screen is visible for everyone.
Yes, ma'am.
Okay. To start with, I have chosen my topic as innovations unveiled, novel approaches to drug design and delivery. Let's start with a simple question. You can feel free to leave your responses in chat. How long do you think it typically takes for a new drug to reach the market? Usually 10 to 15 years. But innovations like mRNA platforms have challenged that timeline. This is my agenda for the today's session, in which we will be covering all the topics that are mentioned. To start with, the transformative approaches in pharmaceuticals. So, as we already told, it would take 10 to 15 years for a new drug to reach the market. But innovations like mRNA platforms have challenged that timeline. For example, the COVID-19 vaccines. Within no time, it started a new era of drug development. The regulatory shift, and about 11 months after the SARS-CoV-2 genome was published, the mRNA COVID-19 vaccine was the fastest, showcasing how new technologies can accelerate the drug development. We are indeed in a paradigm shift as from traditional small molecule approaches to integrated platforms involving AI, biologics, and advanced delivery systems. Likewise, in 2023, there are 7 gene therapies approved by FDA, a record number that signals how rapidly advanced therapies are moving, which underscores a new era where drug discovery, development, and delivery are faster and more innovative than ever before. Coming to the significance and innovation, this is critical to address ongoing challenges from antibiotic resistance to complex chronic diseases. as seen in the COVID-19 response that has streamlined the R&D department. Modern techniques enable precision targeting of diseases and customization of therapies to genetic profiles. The outcome? The patient benefits through better treatments, which arrive sooner with more precise action and fewer side effects. Coming to the real world example, we have an example called halicin. Halicin is an antibiotic that was introduced by AI, which we'll be further discussing in the next slides. In this presentation, we aim to cover all the science techniques that I am currently practicing in our lab and also with cross laboratory techniques. Coming to the next slide, let's go into the artificial intelligence in drug discovery. What do you think AI can do in drug discovery beyond speeding up compound screening? So, what are the other applications of AI other than expediting the drug manufacturing process? AI can predict molecular interactions, suggest lead modifications, and even propose novel drug candidates. AI is revolutionizing how we discover drugs. Machine learning and deep learning models can analyze vast chemical and biological data sets to predict which compounds might become effective medicines. This significantly accelerates the identification of viable drug candidates and can also flag potential failures early. Notably, the first AI designed drug candidate entered human trials in 2020, which was developed by the company Exscientia. Since then, there were 15 AI designed molecules were in clinical development across multiple companies. These AI systems rapidly sift through millions of chemical structures, which takes decades of years if it was humans, even proposing novel ones that humans might overlook. For example, an MIT AI model screened over 100 million molecules in days and identified a potent new antibiotic, which was named halicin, that can kill various multi drug resistant bacteria. This drug worked against some untreatable infections in lab tests, demonstrating AI's power in uncovering entirely new therapeutics. The efficacy of the tools would be to improve the speed and precision of drug discovery by predicting how a molecule will interact with biological targets, how its likely efficacy and potential toxicity would be. AI enhances accuracy in targeting. By learning from big data, these models can design molecules optimized for specific mechanisms. There is also a real world scenario in which the use of DeepMind's AlphaFold, which is an AI for protein structure prediction, which provides accurate protein models, accelerating target identification globally. In our lab, we apply cheminformatics and predictive modeling to rank antimicrobial agents based on bioactivity profile. AI is not replacing scientists, it's empowering them to make faster data driven decisions. The first new antibiotic that AI found was halicin, a compound that showed bactericidal activity against a range of resistant pathogens. In mouse studies, halicin cleared infections where all other antibiotics failed, and bacteria did not develop resistance to it during testing. This was published in 2020, which was the first AI discovered antibiotic, highlighting AI's potential to address urgent health threats in ways traditional methods could not do. Such examples strengthen confidence that AI can help us tackle tough targets like Alzheimer's and certain cancers by uncovering novel chemistry. Moving forward, expect AI to be integrated across the pipeline from identifying targets, for example, analyzing genomics, to finding new disease pathways to designing clinical trials, optimizing patient selection and dosing. The takeaway: AI in drug discovery is transitioning from hype to real results with tangible drug candidates and even clinical trial entries to show for it. This is a typical comparison chart for classical drug development and AI driven drug development, in which there is a lot of saving of time with also lesser side effects and lesser impact or usage of animal models. Coming to personalized medicine. Personalized medicine involves customizing medical treatment depending upon for the individual patient, because one size fits all approach is not the best approach, I feel, because the medicine that works for me doesn't have to work for you, since your genetic and my genetic models are completely different and our lifestyles are different, the foods we eat are different, the gut microbiome that I have is different, and the reactivity or sensitivity that I am to few drugs which you might not be. So personalizing medicine is important. This approach increases the therapeutic efficacy, designed to target that mutant protein, sparing patients without the mutation from unnecessary treatment. By accounting for individual variability, personalized medicines improves outcomes and can transform lethal diseases into manageable ones. Examples include Herceptin. Herceptin is a monoclonal antibody which targets HER2 positive breast cancer. This drug is prescribed only to patients whose tumors overexpress the HER2 receptors, which is just the 20% of breast cancer patients have. So it doesn't affect all the patients with breast cancers. It only affects with the receptors of HER2. The impact has been dramatic. Adding Herceptin to chemotherapy has reduced 10 years of breast cancer mortality by around one third in HER2 plus patients. Breast cancer is commonly seen in 2 out of 10 individuals in USA. So here's the solution. Personalized therapies have been a hit in breast cancer industry. There is also significant improvement in the survival rates. Another breakthrough that is happening is CAR T cell therapy, such as Novartis company has developed a Kymria product, which are custom crafted immune cells taken from a patient. They are genetically modified to attack that patient's cancer and infused back. So this is a personalized cell therapy having achieved remarkable remission rates in leukemias and lymphosis. As of 2023, 6 CAR T therapies have been FDA approved for various blood cancers. However, there are few challenges in this. Despite its promise, personalized medicine faces hurdles. Genetic diversity and data interpretation is one. We are all unique, and not every genetic variant's effect is understood. So it makes challenging for more research time, and sequencing a patient tumor may reveal dozens of mutations. So, but identifying the driver mutations which is necessary for the target is complex. Another challenging is, as we discussed, the research takes a lot of time for each individual. The access and cost also varies. So, however, there are benefits, there are also limitations. Bottom line is, while tailored treatments are revolutionized care, broad implementation will require overcoming cost, complexity, and educational barriers. Ongoing research and health policy work aim to make personalized medicine more universally accessible. recent advancements in microbiology. This is the department that I am working in. So, in current lab, we are mostly focusing on CRISPR-based diagnostics, like those from COVID-19 vaccines, because they are faster and cheaper than PCR. Uh, do you guys have an idea of microbial based treatments that's FDA approved? Feel free to leave your responses in chat if you know, or I'll be discussing it in the session. Rebiota is a fecal microbiota transplant product. It was FDA approved to treat recurrent colon infections. In our lab, we mainly focus on synthetic biology that allows engineered microbes to deliver drugs inside the body. We use MALDI and qPCR for rapid microbial ID, which are critical for infection control and precision antibiotic use. A striking example is how quickly the coronavirus, the SARS-CoV-2, was sequenced in January 2020. The first genome was released online just days after initial outbreak reports, enabling researchers worldwide to start working on diagnostics and vaccines immediately. Today, millions of pathogen genomes, over 15 million for SARS-CoV-2 alone, have been sequenced and shared, helping to monitor an emergence of variants, informing vaccine updates. Coming to CRISPR-based diagnostics, CRISPR is best known for its gene editing tool. Upon finding a target genetic sequence, we will be discussing more detail of CRISPR in the further slides. Coming to mRNA therapeutics, with mRNA, you just change the sequence, not the whole process. So, Moderna and Pfizer's vaccine were built and tested within months after sequencing this vaccine. mRNA is now in trials for the HIV, Zika virus, and personalized cancer vaccines. Microbiome therapies aim to restore a healthy microbial balance. This approach is incredibly versatile. By changing the sequence, you can prompt cells to make virtually any protein, be it a viral antigen for vaccines or a missing enzyme in a genetic disorder. For example, like sickle cell anemia. We just need to change the pattern of the DNA, in which CRISPR is the technology that we use for that. So, mRNA is a transient molecule. It does its job, then degrades. So, it doesn't have any effect on our body, which adds the safety profile. So, how it works is a piece of mRNA coding for a target protein is formulated in a delivery system. Usually, lipid nanoparticles are used for that, which are injected. Once inside cells, the cell's ribosomes read the mRNA and manufacture the protein. If it's a vaccine, that protein is displayed to the immune system, training it to fight the real pathogen. If it's a therapeutic protein, it can directly replace or fix a deficiency. This concept turned into reality with the COVID-19 vaccines. That is the success story of COVID-19, as everyone knows. COVID-19 was the first large scale use of mRNA technology, proving that cells can be directed to create the desired antigen and provoke a protective immune response. This validated the decades of research that went into mRNA delivery systems and modification. It also provided a blueprint for responding to future pandemics. The success has spurred a wave of investment into mRNA platforms. Beyond COVID, mRNA vaccine trials are underway for influenza, Zika virus, cytomegalovirus, and even HIV virus. Notably, companies like Moderna and BioNTech, which rose to prominence through COVID vaccine, have broadened the pipeline to target cancer and other diseases using the mRNA technology. The future of mRNA extends far beyond vaccines for infectious diseases. One exciting frontier is personalized cancer vaccines. In a recent clinical study, a personalized mRNA cancer vaccine tailored from a patient's tumor mutations was combined with immunotherapy. Pembrolizumab reduced the risk of melanoma recurrencing death by 44%. So, a truly individualized therapy is ready for the mRNA. They are aiming to treat autoimmune diseases by inducing tolerance to certain antigens. For example, an mRNA therapy is in development for phenylketonuria. This is a metabolic disorder to provide the enzyme that patients lack. So basically in this disease, patient will be lacking an enzyme. So to replace that enzyme, we are using mRNA. And also cystic fibrosis is a general known example for this technique to produce functional CFTR protein in lungs. The beauty of mRNA is in platform nature. Once you have a delivery method and manufacturing pipeline, you can plug in a new sequence of a new disease relatively very quickly. Regulatory agencies have signaled support, creating clearer guidelines for mRNA productive approval given the COVID experience. Coming to nanotechnology in drug delivery systems. Nanotechnology involves the design and manipulation of materials at the nanometer scale. Nano is a 1 billionth of a meter, roughly 1 to 100 nanometers in size. In drug delivery, nanotechnology enables the creation of nano-sized carriers like nanoparticles, liposomes, dendrimers, and nanoemulsions. These all support for the transport of therapeutic agents in the body in a clever way. Because of their tiny size, nanocarriers can interact with biological barriers at the cellular and subcellular level, improving how a drug is absorbed, distributed, and released. For example, nanoparticles can more easily pass through cell membrane or accumulate in specific tissues like tumors due to leaky blood vessels, a phenomenon known as enhanced permeability and retention EPR effect. The result is that drugs delivered via nanotechnology can achieve higher concentration at the disease site with lower exposure elsewhere, enhancing efficacy, also reducing the side effects. Nanocarriers can target specific cells and release drugs over time. Example, Doxil. Doxil is a liposomal doxorubicin which reduces heart damage while treating cancer. COVID, can someone mute please? COVID-19 mRNA vaccines rely on lipid nanoparticles to deliver fragile RNA safely. In our lab, we are working on polymeric nanoparticles to improve oral delivery of poorly soluble antifungals, which enhances the efficacy. Nanotech also allows combining multiple functions in one package. A single nanoparticle could carry both a drug and a targeting ligand, and even an imaging agent for tracking. It enables us to deliver drugs that were previously too toxic or unstable in traditional forms. The benefits and challenges top. So, nanoparticle delivery systems offer several key advantages, like nanocarriers can be decorated with antibodies or other molecules that bind specifically to certain cell types. Example, cancer cells. Delivering the drug delivering the drug to bind to specific tissues where it's needed most and sparing healthy tissues. So it is very target specific. And it also have controlled release. So drugs can be encapsulated and they can be slowly released from nanoparticles over time, maintaining therapeutic levels for longer and reducing dosing frequency. Improved solubility. Many drugs, especially newer complex molecules, are poorly soluble in water. So, formulating them in nano emulsions within liposomes can make them soluble and bioavailable. The challenges, coming to the challenges, manufacturing and scalability for producing nanoparticles with size, charge, and drug loading on industrial scale is non trivial. Ensuring batch to batch consistency and stability over shelf life requires advanced manufacturing processes. There are also regulatory hurdles. The regulatory agencies like US FDA have well defined criteria for traditional drugs. But evaluating nano formulations, since this is a novel technique, it can be more complex. So, questions about how to characterize the particles, how they distribute in the body, and their long term safety must be answered for the approval. Regulators often ask for extensive data on nanoparticle chemistry, manufacturing, and controls. There are also safety and toxicity issues. While they aim to reduce drug toxicity, the particle themselves need to be safe. So if the particle themselves they are safe, they can reduce the toxicity. If it is harmful, though it reduces the toxicity, it would itself become a toxic. So issues like accumulation of nanoparticles in organs, interaction with the molecular level with other tissues. In long term studies, they need to ensure like inorganic nanoparticle doesn't cause inflammation or doesn't affect the other issues. Overall, the risk benefit equation has to be managed. We weigh the improved delivery and patient outcomes against the complexities introduced. The good news is that as technology advances, many of these challenges are being addressed and regulatory bodies are becoming more experienced with nanomedicine evolution. Example are, as I told, Doxil. It is a liposomal doxorubicin. It was approved in 1995, which is way long back. Even I was not born that time. So it is a nano drug which is encapsulated in PEG lighted liposome. So by packaging doxorubicin in liposomes, it changes the drug's behavior. So it circulates longer time in bloodstream and it accumulates in the tumors via the EPR effect that we discussed earlier. So which means it delivers more chemo to the cancer and less healthy heart tissues. So it has a dose limiting toxicity and the result is effective tumor control with reduced side effects like cardiotoxicity. It also demonstrated the power of nano delivery and has been used for cancers such as ovarian cancer and Kaposi sarcoma. Its success opened the door for many other liposomal drugs like Abraxane. Abraxane is a nanoparticle albumin bound paclitaxel. So, paclitaxel is a potent anti-cancer drug that was originally formulated with harsh solvents due to its poor solubility, causing several side effects. It later formulated and bound to albumin nanoparticles with 130 nanometers in size, which reduced its toxicity, and it has gained approval for breast cancer, lung cancer, and pancreatic cancer with very few allergic reactions since there is no solvent. There are also like other drugs like Onpatro, which is also called Patiserin, which was approved in 2018. Um, it is treated for hereditary ATTR amyloidosis by silencing the pathogenic gene in the liver. There are also some mRNA vaccine lipid nanoparticles which were used in the COVID-19 mRNA vaccines in Pfizer and Moderna. There are also nano emulsions in drug delivery for improving the solubility and absorption. The nano emulsions are itself the tiny droplets of one liquid dispersed in the other. They are used for topical medications. An example is a fenofibrate drug, which is used for cholesterol. It is a nano emulsion formulation which increases the absorption of gut. Another example is topical medications and vaccines using oil in water nano emulsions to boost uptake or immune response. But patients receiving these treatment might not realize it, but they are benefiting from nanoscience behind the scenes. Coming to the regulatory challenges, why do you think some drugs take longer to get approved even if they show promise early? Because regulatory hurdles ensure safety. Even though the drug is effective, they need safety. But novel technologies like AI or CRISPR or mRNA techniques, although they show promising results, uh, we are not sure of how safe they are and in are they safe in everyone. It is not one for all. So, for example, Casgevy, it's a CRISPR based therapy for sickle cell disease. It was fast tracked by FDA in 2023, but FDA approval took more time, although it showed proven results, because it's not easy to approve a drug uh, while showing promising results, since it has to prove safety in clinical trials. The landscape is complex and multi-layered, involving guidelines from agencies like FDA. FDA is for US and EMA is for Europe, and others for the respective countries worldwide. These regulations are in place to ensure safety, efficacy, and quality of pharmaceuticals before they reach patients. Typically, the pathway includes laboratory research, multiple phases of clinical trials, and then rigorous review of results by regulators. In recent years, novel therapies have tested the adaptability of this framework. They have designed like breakthrough therapies and accelerated approval to help promising innovations reach patients sooner, but they must balance speed with thoroughness. For example, FDA Center for Biologic Evaluation and Research now oversees cell and gene therapies, reflecting the specialized oversight needed for these complex products. Global agencies often collaborate, such as Project Orbis, where FDA, EMA, and others review cancer drug to expedite approvals across countries. Still, navigating these processes is challenging for innovation. So, global harmonization remains a challenge. Like one product might pass in the USA, but it might get stopped in the Europe because the regulatory consideration for each country varies. And due to this, the global harmonization remains a challenge. Overall, the regulatory environment is evolving, aiming to keep up with the pace of innovation while upholding the gold standards of patient safety. The key challenge is focused by the innovators. Companies and researchers developing cutting edge therapies frequently encounter several challenges. Lengthy approval process. The classic drug development timeline can exceed 10 years from discovery to approval. Even with expedited programs, it often takes several years to accumulate necessary evidence. This is frustrating when technology is advancing fast. By the time a therapy is approved, science might have moved forward. For instance, CAR T cell therapies had to go through painstaking trial phases, even though early results were dramatic. Maintaining manufacturing consistency and proving long term safety to regulators took time. There are stringent clinical trial requirements, like regulatory bodies require extensive clinical trial data, demonstrating not only that drug works, it also have to be safe, but it also benefits the outweigh risks. Like for innovative therapies, designing these trials can be non-trivial. How do you run a placebo controlled trial for a gene therapy that offers a possible cure for a deadly disease? Ethical and practical issues arise. Additionally, diversity in trial populations has come under focus. FDA now often mandates strategies to include diverse patient demographics, which can be challenging because to gather demographic data from each patient takes longer time, and compiling the data and sending it to approval is infinitive, which can be challenging, but it is important for generalizable results. Evidentiary standards for novel technologies. For example, with gene editing techniques, the gene editing therapies, regulators require long term follow up, like often 15 years for patients, to monitor the delayed effects, which is a much longer commitment. Because for the gene therapy related, the effects might not shown be in the same year, it might take several years. So it delays the monitoring, so which also delays the commitment of approval. So similarly for AI driven process, FDA might ask, how do we know the AI didn't miss something? So that requires explainability or additional validation of AI predictions. So that's the impact on drug approval. The ethical implications of CRISPR technology. Coming to this, should we be editing human embryos to prevent disease? Just give it a thought. The CRISPR-Cas9 is a revolutionary gene. I have given a diagram in which you'll have an idea of how CRISPR works. So it is basically a gene editing tool that allows scientists to make precise changes to DNA in living cells. It's often described as molecular scissors. So the CRISPR is often described as molecular scissors, which is guided by a GPS tool. The GPS is which guides to the location, which is guided by a GPS tool. So a small RNA molecule guides the Cas9 enzyme to a specific DNA sequence, and this Cas9 cuts the DNA at the particular spot. So this enables either disabling a gene or inserting it. So if a gene has mutations, it can either be disabled or it can also be inserted with other repairing gene. The simplicity and precision of CRISPR have made gene editing much more accessible than older methods. Therapeutically, CRISPR helps promise to cure genetic diseases by directly correcting DNA errors. For instance, disorders like sickle cell anemia or beta thalassemia, CRISPR can be used to edit a patient's bone marrow. stem cells to elevate the disease. Even the bone marrow stem cells are used in thalassemia to cure the disease. In fact, in 2023, the first CRISPR-based therapy brand named Casgevy was approved by the FDA for the sickle cell disease, marking an incredible milestone. Patients' own stem cells are edited with CRISPR to produce healthy hemoglobin. While this shows the therapeutic potential, CRISPR is a double-edged sword. Its power raises profound ethical concerns. The ethical concerns are designer babies and germline editing. The most high profile ethical debate is about editing human embryos, which is also known as germline editing, which would result in changes passed down to the future generations. In 2018, a Chinese scientist announced the birth of first CRISPR edited babies. So the babies were born with this CRISPR technology. So they have edited the embryos to make them resistant to HIV. Since the HIV passes from mother to child, they edited the genes of the embryo so that the HIV does not pass from mother to child using the CRISPR edited technology. This announcement was met with the global outrage. It was meant as unethical and uncultural to do in such way for a premature baby without sufficient transparency or oversight. Although it was successful, there were ethical concerns for that. The fear is that it could be used not just for disease prevention. So knowing this technique, people might misuse. They might use misuse for like non-medical trait selection, like editing the genes and making the babies however they want. So these led to the concerns. So these were termed as designer babies, and host of social and ethical issues like inequity, eugenics. So from that time, it was since called for a moratorium on clinical germline editing, and many countries have laws banning this technique since the baby incident underscored how critical global ethical standards are. It's a cautionary tale that just because we can do something doesn't mean we should. Just because it's possible doesn't mean that we should do something, at least not with broad consensus and regulation, and at least not with the societal. The future directions. The future directions. So imagine this: AI designs a molecule, CRISPR corrects a mutation, mRNA delivers the therapy, and a nanoparticle ensure it reaches the right cell. It sounds futuristic, right? These are addressing medical needs for like all the diseases, all the genetic diseases. Most ethicists agree that somatic editing for treatment with informed consent and proper review is ethical. So, the general consensus from global summits on human genome editing is that it's too early to safely and ethically edit CRISPR technology. Artificial intelligence and machine learning. AI will become even more ingrained across drug development pipeline. We anticipate AI not only helping discover the new drug candidates, but also personalizing treatment plans in real time. For instance, AI algorithms could analyze a patient's medical records. Can you imagine? And genetic data. So there is no need of nurses who can drive the data. It's just all AI. And they can even monitor the data to suggest the drug dose that you need to take. It's a truly individualized therapy. AI driven design for proteins, antibodies, they could lead to bespoke therapies for very specific targets. We may also see AI helping to predict and mitigate side effects early by simulating how drugs influence complex human microbiology. mRNA and beyond. As techniques improve, the precision will increase and the spectrum of treatable conditions will broaden. Imagine editing immune cells on the fly to combat a new virus or correcting a disease mutation shortly after birth to prevent a child from developing a genetic disorder. That would be so good. The success of mRNA for the next generation vaccines will likely tackle cancers, autoimmune disorders, and tricky infections like HIV. Additionally, cRNA and antisense oligonucleotides, which modulate gene expression, are gaining traction. The approval of mRNA drugs like Onpattro signals more to come. The cRNA injection twice a year that silences the gene making LDL cholesterol, complementing or even replacing daily pills. So, people who had the LDL cholesterol no need to take pills daily, so they can silence the genes that makes the LDL cholesterol. Sounds interesting. Regenerative medicine and cell therapy. They're also used in the regenerative medicine like tissue engineering. AI and tissue engineering can also be used for agriculture. It's not only have applications for clinical trials, but it can also be used for agriculture and it can be used for specific tissues like pancreas and diabetes like Parkinson's disease. So these therapies blend engineering, biology, and future drug delivery. So this may involve supporting these life implants or ensuring they integrate properly. Also, I have a note on organs and chips in silico trials. The future drug testing might involve a suite of human organ chips, which are like heart, liver, kidney, to, to quickly evaluate the safety. So rather than having clinical trials on the whole body, they might be like organ specific, like specific for a human heart or a human liver. So before moving into real humans, they will be testing on the human heart models or a liver model or a kidney model before moving into human models so that the loss is less. So combined with AI, this could streamline development and allow more iterative design of compounds. Quantum computing. This is also more speculative, but quantum computers could eventually solve complex molecular simulations that classical computers cannot, possibly revolutionizing drug discovery by accurately predicting drug target interactions and guiding precise design of molecules for tough targets. In summary, the key takeaways of today's presentation would be the toolkit of future drug designers will be rich, like AI, advanced genomics, novel biology platforms, and powerful computing. The traditional boundaries between disciplines will blur. So, biologists working with data scientists, chemists with AI engineers, and fostering cross disciplinary innovation. The sustainable and ethical practices. As we embrace the innovations, there is a parallel push for sustainability in pharma. Drug design and manufacturing are being rethought to reduce environmental impact. This means green chemistry. Using environmental friendly reagents and processes minimizes waste. So it also reduces effect on environment like low waste packaging, decentralized manufacturing, which will drive the next decade of pharma. Big pharma companies like Pfizer, Novartis, they have already implemented these green chemistry programs to redesign synthesis of drugs to be cleaner and more efficient. This not only benefits the planet but also lowers the cost. So this is like eco-friendly manufacturing, like recycling water from manufacturing or using biodegradable plastics, using less water and energy. They're exploring renewable energy sources. Uh, Ethical sourcing, ensuring that the raw materials like plant-derived compounds, uh, which includes the medicinal chemistry or rare minerals used in catalysts, they are sourced responsibly and without depleting natural resources or exploiting communities. Coming to supply chain resilience, the COVID pandemic taught the world about supply chain vulnerabilities. Future drug delivery will involve more robust, perhaps decentralized manufacturing, like portable manufacturing units that can be quickly set up near the point of care, like producing the dose for personalized therapy. This can reduce the transportation emissions and respond faster to the demand. Collaboration and continuous learning. A key thread through all the future directions is that no single field can do it alone. So, collaboration between science, technology, and regulation is essential to unlock the full promise of these innovations. Researchers will need to work hand in hand with tech companies, like for AI, machine learning, and with devices and with regulators from early stages to craft acceptable development pathways. We have seen examples of this with FDA's collaborative communities on topics like AI in medical devices, similar models that may extend like AI in pharma. For professionals like us and all the audience, the message is clear. The key to progress lies in continuous learning and adapting. We must stay curious and open minded, ready to update our skill sets. For instance, a biologist or a chemist learning some data science, a chemist getting familiar with CRISPR biology. Like cross disciplinary thinking will span the next big breakthroughs. Maybe a nanotech expert partnering with an immunologist yields a new targeted drug delivery capsule, which can train the immune system. So, academic training itself is evolving to be less siloed to prepare the next generation. So, who are as comfortable with a pipette as they are with Python coding script? So, I feel continuous innovation is imperative. Coming to the conclusion, the integration of disciplines, the microbiology led innovations, we have discussed about the AI and CRISPR technologies that are transforming the drug discovery, delivery, and diagnostics. What began as experimental tools, and there were just the laboratory experiments that now have come into practice. We are using those techniques, which were now as models, like they have become the smarter therapies. As already discussed, the key to progress lies in continuous learning and adapting. So, upskill yourself, cross collaborate. If you are in pharma, also show interest in learning data science or Python, since you never know where you'll be ending up. So, I encourage all to have involved in all the disciplines and have responsible adoption, because with great innovation comes great responsibilities. Ethical and regulatory considerations should be viewed as the frameworks, not impediments, because we guide innovations to be safe, equitable, and beneficial, builds public trust. Stay adaptive and curious. Perhaps the most important trait for anyone in this field is adaptability. What is experimental today may be routine tomorrow. So, being will to continuously learn, whether it's a new programming language for AI or a new protocol in gene editing, that will position you to lead and not just follow these advancements, also adapt to it. The coming years in drug design and delivery promise to be exciting and transformative. But by staying informed of global developments, collaborating widely, and keeping the welfare of patients and society at the center of our efforts, we can ensure that these innovations realize their full potential in improving human health. So the closing line would be change is constant, and those who learn lead it and also adapt to the change. These are the references and further readings if you are interested more. I hope this session was not boring. It was most insightful I could make it. And please feel free to leave any responses, questions, or concerns in the chat box. Thank you for all your time and attention today. I hope this session offered a meaningful look into the cutting edge innovations driving modern drug design and delivery. I hope at least one information is useful and you have learned something new. It was a truly honor to share all my insights drawn from real world projects, current industry trends, and my work experience. I would sincerely thank all the organizers, the principal sir, and everyone who have participated, took time in this weekend. So please feel free to reach out to me if you have any questions, would like to explore any of these topics further or in detail, or need guidance related to your careers in pharmaceuticals, regulatory affairs, or the lab sciences. Please feel free to reach out to me. Wishing all of you a continued growth and success in the academic and professional journeys.
Thank you, Shalini ma'am.
Thank you so much.
Thank you, ma'am, for sharing your valuable knowledge about various techniques used in the drug design process, which includes AI technology, recombinant technology, and nanotechnology, as well as thank you very much for your valuable insight about regulatory challenges in the drug design and discovery process. Thank you. Some more questions are there in chat box, ma'am. Please.
Oh yeah, sure.
Questions.
Just
One question, ma'am. This type formulation stable or not in journal atmospheric conditions? From one participant.
uh, okay. Where is the question? Sorry, I didn't see. There are so many.
In chat box only, ma'am, at
Okay.
10:14.
Oh, these type formulations stable or not in zonal atmospheric conditions. So yeah, these type formulations are, I would say, it would be diplomatic. They might be stable. They are in atmospheric conditions. It depends upon the properties of the product. So it is a diplomatic answer. It might be stable or no. It depends upon the properties that we are using for the drug.
One more question, ma'am. Can we use AI for UG and PG level innovation research, like creating research protocols?
Yes, definitely. You can use, and yeah, you can use, but not copy it. So use the information that AI is providing and give your own thought and modify it. It's not that you just copy from the AI. You also have to give your own mind to it and write the information. You can use AI for research.
Dear participants, if you have any queries, please ask them.
Good morning, madam.
Yeah, good morning.
Regarding, for example, a person is suffering from this palindromic means abnormality
Okay.
in like a Philadelphia chromosome abnormality is there. Right?
Mm-hmm.
So how do you identify that? By using this technology and all, you have told by using CRISPR technology, we can modify like that. You're saying, right?
Yes.
So, for example, the person is having this problem called as Philadelphia chromosome abnormality. So, especially we use a drug called dasatinib for that. So, how do we identify that abnormality in that DNA? And
So-
how we can overcome that abnormality by using these technologies like CRISPR?
Yes. So thanks for mentioning. So the CRISPR technology, how we use is the DNA has a specific sequence in which the strands have the mutations. So that particular chromosome which is affected would have a sequence in which it got affected. So using the CRISPR technology, a guided mRNA will be attached to the CRISPR technology, which is used as a protein, and it is sent into the body. When it is sent into the body, it only identifies which we give a the mRNA is located with the CRISPR edited gene, and it will be located where the chromosome has to be replaced or substituted, or whether it have to be inserted or taken out. So when it is identified, the CRISPR cuts the necessary tagline or the chromosomal edited DNA and replaces it with the necessary DNA. So it is guided with a protein. So it identifies the chromosome that has defects and replaces it or substitutes it.
Okay, madam. Like you are saying, we can cut the DNA, we can insert the new DNA like that. Doesn't
Cut
that
the
affect
part
the?
of DNA.
Yeah, I understood. But
Yeah.
doesn't it affect the person's means natural immune system or
Genetic.
genetic hereditary material?
No,
Does it have
it
an
doesn't have.
abnormality?
No, it doesn't have any effect on the other healthy tissues. It just affects the mutated genes. So the CRISPR technology is safe in that way that it doesn't affect the neighboring tissues or the healthy tissues. It only affects the tissue that have been targeted. So it is very target specific.
Okay, ma'am. Thank you.
Thank you.
Shalini,
Sir,
very informative section, Shalini.
thank you so much, sir. Thank you for this opportunity.
I feel very proud at this moment, Amma.
Thank you so much, sir. It's all of your blessings, sir.
Very good, Amma. Keep going.
Thank you so much, sir. I would like
Okay.
to give a special thanks to Balakrishna, sir, for providing me this opportunity. He was the primary reason that have motivated me as a student, and I am here now. Yeah. He never made me to give up for anything, whether any seminar or anything. That made me to give a seminar by myself. Thank you so much, sir. I think you are muted, ma'am.
Thank you once again, ma'am, for accepting our invitation in spite of your busy schedule. Thank you. As a
My
token
pleasure.
of appreciation, on behalf of University of Pharmaceutical Sciences, we would like to present a certificate. Please accept
Thank
it.
you. Yeah, sure. Thank you so much. It's my pleasure to be part of the team, and thank you. Thank you so much. And yeah, please feel free to reach out to me for any career guidance or opportunities or anything. Thank you so much. Thank you.
Thank you
you,
once
ma'am.
again, ma'am. Thank you. Dear participant, attendance link is posted in the chat box. Please fill the form by today evening itself. Thank you once again. Thank you, ma'am.